Thank you so much, Dr. Serrano and Dr. Simpson. That was so wonderful to hear. And for the next 5 to 7 minutes, I'm going to highlight some of the questions from the community that I hope you'll be able to answer. So it looks like our top question is about CRISPR. Dr. Simpson, the question is, I've heard that CRISPR is hard to apply to the KMT2 gene because it's so big and the spectrum of mutations is so broad. What's the reality of being able to use CRISPR in human KS patients? Or is that just a quick method to build iPSC cell lines?
That is a great question, and Dr. Serrano may also have some insight as well. But I will say at this point in time, CRISPR is an amazing technology, but is the new kid on the block. And so in many respects, it's not quite ready for primetime patient care. And so, and I mean that because you're right, the KMT2 gene is extremely large. It's very, it can be more difficult to work with, but then also part of the inherent problem with CRISPR and utilizing it in patient lines and in patients, is that you just have a little 20, you can think of 20 little letters that are kind of on it, and it's guiding and going through, and it's finding in our large book with millions of little letters where to pop down. And we see in the lab that you have to really specifically pick which clones you use, and you have to not use all of them because there are off-target effects. That means that when we give CRISPR, it doesn't just fix or change that one spot. It actually can go in and change other ones because they only have 4 letters in our book to work with. So there's going to be overlap other places. It's not as specific as I think it was touted to be initially, but it still is incredibly powerful. And we're getting a lot better at utilizing it. It's just not ready to be given to our Kabuki patients yet. And so it is a really great method to quickly build iPS cells. And you can utilize control lines, which in the biology world is really important, just to make sure what you're seeing is what you're actually looking at and not one of those off targets. So
thank you so much, Dr. Simpson. One of the next questions that I think is really relevant to our overall efforts at the foundation is this idea of collaboration. So this question, again, is for Dr. Simpson, but Dr. Serrano, I'm sure you have a perspective as well. Dr. Simpson, when you say you're working on the natural history of the disorder, it seems like other institutions are doing the same, like BCH, Manchester, Kennedy Krieger. Is all of this data being aggregated and shared across institutions? And are you measuring all the same things to ensure that there's standardization?
Is a great question. And so, yeah, the natural history study on our end was started prior to us joining the endeavor through the Kabuki consortium that we're building. But absolutely, the data that we're collecting is going to be utilized by everyone within the consortium. And that's a great question. We're working to really standardize things at this point. But right now, at least the measures that we're using, we've got some behavioral assessments and things that are standardized, but we do have some things we're asking differently, but we're working really to kind of standardize it across all the centers, just because we know that families have a finite amount of time and we don't want to waste your time with a bunch of different questionnaires and things. And there's going to be more actually at the conference that you guys are going to hear about too, that will help streamline some of that too.
And that's a great moment to just mention the foundation and some of the work that we're doing. So often institutions are really deeply involved in driving the research that they're responsible for. And it's important for a patient-centered organization like the Kabuki Syndrome Foundation to come into the fold and help drive collaboration and help fund opportunities to make sure that things like natural history studies are usable, standardized, that the right documents and agreements are in place across these medical institutions to share data. So one really important way to make that happen is to donate to the Kabuki Syndrome Foundation. You can go to kabukisyndromefoundation.org and engage your families and community to help drive forward these types of efforts.
And
Dr.
I would also like
Please.
to add to that is that by helping this initiative by the Kabuki Syndrome Foundation, we are setting standards and map roads for any other rare disease with similar challenges. So it's not just a one collaboration for one foundation. It's something that we're looking forward to synergize with different foundations. So it's extremely important.
Absolutely. Cross-institutional collaboration is top of mind for us at the foundation, and I know it is for all of the researchers in our consortium as well. Dr. Serrano, a question for you is this idea of spending time within your community. Have you seen specific patient-driven action or changes in research as you've continued your work?
Well, if this question is particular of the Kabuki syndrome community, I just share a little bit of my own experience after sharing these gatherings with the families and talking with them, and they asking me questions about this, for example, memory recall that we heard from one of the questions. Learning about all of that, all of those elements were key for me to drive and to shape my research project. So those efforts, as in my case, I don't have contact with patients if I don't look for it, if I don't seek for it, because I'm a basic scientist. So it's super important that the community also opened the door to people like me, for example, who wanted to learn from all of you. And I can see this also being contagious. We are seeing more of this in the Cat6A community, which is starting on this effort. So I would say that, yes, I can definitely see, I experience it myself, and I can see it in different foundations.
Amazing.